and welcome to Soapbox Science London. Thank you all for joining us, and we've got 11 amazing speakers for you today. Hopefully you've had a chance to look at everybody's videos on YouTube, and you've got some questions prepared. So if you've not sent your question in advance yet, if you comment them on the Facebook link, then we can see them and we can ask them for our speakers. So to start with, I think what we'll do is we'll ask a question for everybody, which is, what is the thing you enjoy most at work? So do we have anyone who'd like to start, or shall I pick somebody? No volunteers yet. Let's start with Jessica Chandra. Would you like to start with what you like most at work?
Sure. Hi, can you hear me?
Yeah.
I think the best thing I like, so I work in the lab, and the best thing about me working in the lab is that my lab is based in engineering alloys, and there are people working in all types of alloys and all type of industries and applications, and it's really nice to be working. So I work in biomedical field, and to be working next to someone working in like aerospace or like nuclear energy, and I think it's really cool to kind of learn together, even though we have different applications, but the science is the same and we can still talk about it.
It's amazing. Really nice that you've got so many colleagues around you. How about Dr. Inga Asher?
Hiya. You can hear me okay?
Yeah.
I think it's trying to crack a problem. So being able to go into work, try and ask a question, and then over time, not every day necessarily, through successes and failures, try and answer that question. It's really exciting when something works, sad when something doesn't. It's an emotional roller coaster, but it's never dull.
Nice. And remind us where it is you work.
Oh, sorry. I work at the Cancer Institute at UCL.
Great, thank you. And how about Eleanor Crane?
Yes, I also work at UCL. I do quantum physics. And I think what I find the most exciting about work is the freedom that we have to choose a project which is really important to us. And on top of that, we also have the freedom to be able to work with friends if we choose to. And finally, an extra bit is that it's always nice to travel a little, and conferences allow you to travel.
Amazing. How about, I know Heidi, you've also done quite a lot of traveling in your work. Would you say that's one of your highlights as well?
Yes, definitely. So yeah, definitely one of my favorite parts of my work. I work at the Institute of Zoology at the Zoological Society of London. So I work both in the office here, surrounded by very like-minded colleagues. We often have some stimulating discussions, but also a lot of my work is in the field, which is in China for the most part for my project. So I really love meeting different people, and we work across with researchers, with nonprofits, with government bureaus, with local people, and really going to different places, seeing the environment, seeing the species, and talking to different people is really rewarding. And yeah, I would say that's definitely a highlight of my job.
Amazing, thanks Heidi. And how about Dr. Mine Olu?
Thank you very much. I very much colleagues actually. I'm also working at UCL School of Pharmacy, and I really enjoy on a daily basis the meetings with my research team. Actually, I can't wait to meet with my PhD students, which is amazing, which is always exciting. Even they knock my door and say that, Mine, this is something that we weren't expecting to see, but actually any unexpected result actually is interesting, because it leads us to the next stage in our research projects. So they really make my day. This is the highlight, of the day. And in terms of, the meetings with colleagues, with project partners, it's always very interesting to exchange knowledge.
Great, thank you. How about Dr. Vanessa Sancho-Shimizu?
Hi, hello everybody. I'm Vanessa Sancho-Shimizu, and I'm based at Imperial College London. So I think I echo a lot of the thoughts that the others have brought up. I think the most exciting thing for me is the people, coming to see the people that I work with every day, my colleagues, my team, but also I am part of many large international collaborations, and thanks to Zoom and all this virtual meetings, I I get to meet with people from all over the world basically every day, which is fantastic. It doesn't replace the travel, which I must say was a huge highlight as well. But also interacting, just like what Mine said, is I'm really excited to talk to my team every day to see if there are any new discoveries or any new updates. That's what keeps us going.
Thanks, Vanessa. How about Laia Delgado Calico?
Yeah, for me, I work at King's College London in the physics department, studying metallic nanoparticles. And for me, the best thing is learning new things and then getting to share what I've learned with the scientific community and also with society through events like this one.
Great, thank you. Just to say thanks, we've got a lot of questions coming through already on Facebook, so keep them coming. We'll get to individual questions in a minute. But first, maybe Dr. Zoe Waller, would you like to tell us what your favourite part is about your work?
Hello, I'm Zoe and I work at UCL School of Pharmacy alongside Mine. And I really love my job and I really love the variety. So I do lots of different things. So I have a research group who I enjoy interacting with, but also teaching and being able to do things like this and help other people on their scientific journey. So it's the variety that I really like most about my job.
Thanks, Zoe. Sounds great. How about Sheila Barasa?
Hi, thank you. My name's Sheila Barasa. Oh yeah, I can see myself. Yeah, so I'm a PhD researcher at the Barts Cancer Institute, which is a part of Barts Medical School. And my PhD project is in cancer research, of course. And I'd say the best part about what I do is I think it's being able to brainstorm ideas and not necessarily is this always done in a boardroom? Sometimes it's through social events. We'd just be having a barbecue, we'd be sitting by the bench, having a couple of drinks, and someone comes up with this great idea. And this happened last year, and this year, one of my colleagues ended up publishing a paper just from a random idea he came up with. So I think being able to brainstorm ideas and being able to follow them through, I really enjoy that most.
It's amazing. It's a great way to work. How about Dr. Kiko Mizuno?
Hi, I'm working at King's College London of basic and clinical neuroscience, and it's focused about the memory. I'm working on an animal model, but I found things try to find out how can apply to the human problem of memory. That's enjoy I most, and also travel world for meeting to meet old friends, to catch up and to warm up friendship. That's quite enjoying.
Thanks, Keiko. And finally, can we hear from Helen Muller, please?
Hi, yes. I'd say without doubt, one of the best things is the amazing places that I have got to go to through my PhD and other work in conservation, partly through the Zoological Society of London where I work, but also beforehand. And as well as meeting or like going to these really wonderful places, you meet a whole bunch of people from really different walks of life. And it's really interesting putting yourselves in different people's shoes.
Thanks, Helen. Tied into that, I think we're going to jump straight into a question from Facebook, which someone has said, "Hi everyone, thanks for all the great videos. I have a question for Heidi. How often do you travel to Hainan to see the animals you study in the wild? And what's your favorite Hainan animal?"
Good question. So prior to the pandemic, I would be based in China several months a year, both for my own PhD research, but also prior to starting that as the project coordinator. So I'd say the last year in 2019, I'd spent probably 5 months in China, and I will travel to Hainan most of like about once a month, I'd say, on average, and either spend anything from a couple days to a couple weeks to the whole month there. And in terms of the species, my favorite ones, I would be obliged to say Hainan gibbon because that's the main one I'm working on. But recently, a new part of the work that I've been involved in is about turtles, and I've really loved seeing turtles in Hainan for the terrestrial and freshwater ones. And there are about 10 species of native turtles that are all really different, really fascinating. So I would say, other than the Hainan gibbon, I'm really interested in the turtles of Hainan right now.
Oh, amazing. And your drawings of the turtles were really amazing on your video as well.
Oh, thank you.
If I could swap to a question for Jessica. We've got one that was sent in advance, which said, would a titanium implant last your whole life if you got it at a young age, or would it need to be replaced?
So that is a great question. And actually, so the aim is so that we don't have to replace implants, even if you put them if you're slightly younger. So things that will last maybe 30, 40 years. So this is something that's still being actively researched. So I specifically study the corrosion of these implants in your body to, so hopefully in an ideal world, there'll be no corrosion, and therefore it can last in your body for a very long time without any negative effects, and it won't, release particles into your body and things like that, which has happened in the past with other types of implants. But yeah, so the hope is that that won't be the case.
Great. So is it a research phase at the moment? And yeah,
it is, yes.
Great. And how about one for Laia? We've got one for you, which was sent in advance, which is, how do you study nanoparticles and how do you measure something that's so small?
Yes, that's the question. So in my case, I'm not an experimentalist, I'm computational. So I just use my computer and I model, we know the equations that they follow. So we solve math equations with a computer, but you can also do it experimentally. If you have a piece of the metal that you want to study, like a piece of gold, then you shoot a laser and it breaks that piece into small particles that are the nanoparticles. That's how experimentalists do it, but I don't do it myself.
Great, thank you. I'm going to circle back to Jessica again because a question has come in, I think related to your last answer, saying, do you think 3D printing could be used to make organ transplants in the future as well as bone transplants, which you're researching?
I think it's definitely something that's possible. So I'm actually working with a startup company called Alloyd based in Oxford, and their goal is to build custom-made implants. And it's not really particularly specific to any parts of the body. So I heard that in the summer, they're gonna try on a person to replace someone's ankle. And that is kind of the first step. So theoretically, bone and orthopedic implants are slightly easier to manufacture because you can mimic the structure of bones. But things like spine and other parts of the body, I think it's definitely in the future something that we want to do as well, so that we can kind of replace needing like transplants from other people and just manufacture them ourselves.
Great, thank you. That's really exciting. We've got one as well for Zoe, which was sent in advance, which says, could you please tell us more about DNA nanotechnology that you mentioned in the video? Are there any examples where it's used?
That's a really good question, and there's lots of different examples where DNA is used in nanotechnology, and the use of DNA in non-biological circumstances is actually quite widespread. For example, even Microsoft have started to use DNA to store data, so storing information rather than using traditional methods. But in terms of nanotechnology, so we've actually developed triggers within my research group to actually change the shape of DNA so that it can be used as an on-off switch. So you would then have a trigger and it would switch on, or it would have another trigger and it would switch off. So these can be used in like mini logic gates, which are like small computers.
Wow, thank you. And we've got another one coming for you just now on Facebook, which says, why are plants' DNA sequences so much longer than humans? Are they more complex organisms?
That's a really good question as well. And plants are more complicated. If you think about it like with a plant compared to an animal. So if we are thirsty, we can go and locate some water. Whereas if a plant needs more water, or if it doesn't have enough water, it needs to have all of the signaling to detect that there's not enough water, but then also to then grow roots and actually do all of those things. So plants can't move, so they have to actually change themselves to adapt to their environment a bit more than animals do. So that's the reason why their genomes are quite complicated.
Wow, that's really interesting. Thank you. And we've got one for Ella. Do quantum computers look similar to regular computers, or are they very different? How many of them exist at the moment?
So they look relative. So you interface with them via a classical computer. So in the video, actually, you can see how I go onto online, and with the IBM Quantum Experience, you actually have access to like free quantum computers, which are there. You can send them your algorithms. So you can interact with them just by placing, as Zoe was just mentioning, logic gates on all of the different qubits. And what was the second part? Do they look different also actually in that?
Yeah, but how many are there in the world?
Oh yeah, how many are there in the world? It's a good question. I think there's about 3 or 4 implementations of every different style of quantum computer, because you can have quantum computer in the solid state in a real crystal. You can also implement a quantum computer just in atoms and molecules, which are trapped with light beams and magnets. And you can have a quantum computer which is made with superconducting qubits, which is again made with photolithography. So it's a lab maybe similar to the one where Jessica Chandra works. And so there's maybe in total around 10 or 20 quantum computers in the world. It's difficult. There's only about 10 qubits, but in the future, there should be more qubits and more quantum computers.
And you've got one at your workplace as well?
Yes. So at IBM, they have actually 10 quantum computers already. And in the workplace where we have just a few qubits, and we're trying to control them and trying to prolong their lifetimes, which is one of the very, very long-standing problems which we have in our platform, Silicon. Yeah.
Thanks, Ella. We've got one for Sheila. Quite a broad question. What has more of an effect on someone's health and lifespan, their DNA or their lifestyle?
That's a very good question. I think it depends, but I'd like to say the majority of diseases are more lifestyle based. And that's simply because if your DNA has more of an effect of your lifespan significantly, you'd probably die earlier, you'll be selected against, and then you'll be able to say live on, reproduce, and propagate that DNA onto the next generation. So it does tend to be, I think it's the classical kind of nature-nurture debate at the end of the day, but I'd say lifestyle has a lot more of a tangible impact. However, when it comes to DNA, if it's a very, the thing with the DNA is if you do have a health condition that's caused by genetic defect, it tends to be a lot more severe. So it's smaller quantities, but more severe with their DNA. However, with lifespan, that tends to be a lot more common, but that tends to happen more frequently.
Great, thank you. While I have you there, I think I'll ask one that we got sent in advance, which is, please can you tell us more about what you do in your day-to-day work at Bart's Cancer Institute?
Okay, yes, so that's a good question. So I guess I can speak for all panelists here to say that my day-to-day work greatly varies. No 2 days are the same. And because, so my PhD project has been, 2/3 of it has been mainly computational, and 1/3 of it has been in the lab. So I'd say for the past 2 years, my day-to-day would mainly be go into the lab. I tend to have, there's normally a meeting going on each day. It's either a department meeting or supervisory meeting where we just all convene and someone presents, shares their work, do some troubleshooting, you get questions asked. So you do expect to have at least that kind of a meeting. I'd say on average, you'd get that at least 2 or 3 times a week. The more senior you are, it's more likely the more meetings you are attending. And then I tend to have supervisory meetings as well, 1 of those a week. I tend to have a group meeting just for my lab group. So you have a group that you're in could be anywhere between 6 to 12 people. And then you have a department that you're in. So I'm in the Center for Genomics and Cancer Biology. And then you have the overall institute that you're in as well. And then I spend the rest of my day on my computer, and that's if I'm doing some data analysis. However, now that I'm doing a lot more of the wet lab, I could go in on Monday, I'd grow some cells, and then I'd probably start setting up my experiment on Tuesday, and then I'd carry out my experiments for that on Thursday or Friday, doing things such as a qPCR. Great,
thank you. I think we'll go back to a question that we had in advance. Let's have one for Vanessa. Can you predict if you're going to have a serious infection from something like a coronavirus?
I think so it's a complex question. I think we know now that there are certain risk factors that place you at higher risk for developing critical disease with SARS-CoV-2. And those 3 factors are age, so people who are older, your sex, so males tend to be more affected, and various comorbidities are also associated with more critical disease. So those are three factors that we know quite clearly can impact your risk of critical disease. Other than that, I think it's quite difficult to know. And the kind of work we do is looking at sort of the genetics underlying the most critical form of disease. And what we did find is actually there are amongst the most critical cases, up to 3.5% of them carry damaging mutations or genetic changes in their DNA that basically make a very important antiviral pathway non-functional. So we know that actually genetics can play a role. This is only 3.5% of the critical ones. In addition, though, we also found that up to 10% of critical patients also carry these rogue antibodies. And these happen actually, so these people carry autoantibodies to this antiviral molecule that's protective and basically make it completely non-functional. And so we know that if we could carry out additional screening in hospitals upon admission to see if these patients carry any of these autoantibodies, or the genetic screening is a little bit more practically difficult at this point. But the autoantibody screening is something that can be technically implemented and can actually change the course of treatment. So there are various factors that can actually give you an idea if you are at higher risk of developing critical disease with SARS-CoV-2. That said, all those things, there are many other factors that we don't know about or don't fully understand that contribute to that as well.
Thanks. And then we've had a question on Facebook come in, which I think is related, which is how are the clinical trials going for the COVID treatments you mentioned in your video? Is that the same thing you've just been talking about?
Yeah, so I think there are many clinical trials for various drugs right now. I actually am ashamed to say I haven't kept up with all of them. There's a multitude of them, but for the one that's affecting the antiviral interferon treatment, it's still underway. So I don't think that we have any results yet.
Great, thank you. I think we're going to go back to Jessica quickly. Someone's asked, how do you print a metal? How do you 3D print a metal?
So how we usually do it is with a process called laser bed fusion. So we have tiny metal powders, and you melt it with a laser, and you create like a melt pool, like a liquid pool, and then you melt very specific areas with the laser so that you can create a shape that you want in 3D. And usually the powder sits on a platform, and every so often the platform will go down a little bit, and then more powder will be swept into the area. And then that's how you make like a vertically 3D printed shape.
Oh, great, thank you. And then we've got one here for Helen. someone said, I thought your video was really interesting and hadn't considered this perspective on trophy hunting before. If the ban on trophy hunting in Botswana continues, do you think future generations of people will continue to oppose the ban, or could they just adjust to this new way of life?
That is a very interesting question. And I'm glad to hear that you hadn't considered some of the things that I raised. The ban has already actually been lifted. So it was in place from 2014 to 2019, and it was lifted early, or sort of like mid-2019. But quite interestingly, the hunting season didn't actually happen in 2020 because of COVID. So it's only, I think, restarted again this year. But in terms of sort of people just adjusting, getting used to things, I think that'll be quite hard to do. Partly, I mean, just based on my experiences from living in these two villages is that elephants are really quite scary and big and really difficult to live around. And so just those like very real changes to people's lifestyles would be something that does stay with you. So unless there are other ways that can be used to sort of address particularly like human wildlife conflict, then I think you would find sort of people resenting those kinds of decisions, particularly if they weren't made with their consultation.
And just off the back of that, what kind of other ways are there to manage that type of conflict?
Oh, that's really hard. And there are no great ways, particularly when it comes to things like elephants or animals like elephants, which are really clever. So there are all sorts of different interventions that are being done all over the place in Africa and also in Asia, including like loud noises to scare them off. But then as soon as nothing actually happens, the elephants just come back. But also things like making what's called chili bombs. So making sort of growing chilies and mixing them with elephant dung and burning them, that apparently keeps them away for a while. Bee fences, there are all sorts of different ways, and they have varying levels of success.
Great, thank you. I think I'll ask you one as well that we had in advance, which is, do you think decisions about banning trophy hunting should be specific to different areas, or are there some things that you've found in your research that could apply to most places?
That is another really great question. I would say in most cases, it's very context specific. So for some of my wider work has been to look at trophy hunting across Africa and the different evidence for its impacts. And one of the things that I found is that it's incredibly varied, with some countries generally managing trophy hunting very well, where it isn't impacting wildlife populations and it can really do some good to communities, bringing in income. But in other areas, particularly where sort of like governments are quite, or like the regulations are quite weak, then it can be detrimental to both wildlife populations and people rarely involved actually. So you don't actually get this income coming to communities, and they just like having to deal with the costs of living alongside wildlife.
Great, thanks, Helen. I think we'll move on to one for Mine. Hold on, where have we got it? So we had one in advance, which is it just the liver and kidney that affect dosage, or do other organs play a role as well of medicines?
And that's an excellent question. I would like to thank them actually giving me the opportunity to further elaborate on this topic. It's not, unfortunately, only the liver and kidney, which may affect, how the body treats the medicine. And there are other organs as well, because I only mentioned kidney and liver because these are the main organs, which are taking action in the metabolism and elimination of the medicine. But at the earlier stages, after the medicine is taken, gastrointestinal system, for instance, is making an impact if the medicine is taken via oral route. Let's say, if the patient is taking a tablet, a capsule with a glass of water, it first travels to their stomach and to their small intestine, and it travels, to go to the systemic circulation to show its therapeutic effect. And due to aging, if the absorption levels are different, if basically our tissues and cells are taking up the active part of the medicine, which we call drug, which is active pharmaceutical ingredient at a different level, then it will also change the performance of the medicine on an older individual.
Thanks, Mine. And there's one that kind of fits in with that is, do you work mostly with elderly people? Are there any other groups that need patient-centric medicine?
This is another great question. I would like to thank them for highlighting the importance of patient-centric medicine for different special populations. So it's not only the older people that we put our efforts for making, individualized, personalized therapies, but we also would like to answer the needs of many other special populations. So when we say special group, we may refer to a group who are suffering from a certain disease condition, or we may even consider, female patients and male patients, because we know that there are sex and gender related differences when we respond to drugs. So we try to understand, what are the inter-individual differences, how we can maximize the therapeutic response in these different patient groups. So age is another factor. So we try to look into the needs of pediatric patient group as well as older people. We also try to understand the progress of the diseases, because unfortunately some of these diseases are progressive, so they do not show a very stable pattern, and the patient's needs may change. Actually, the individual patient needs may change during their treatment. So we try to address all these changes and try to help actually with our design of medicine approach in the best direction.
That's great, thank you. We've got one for Keiko, please, from Facebook, which says, why do some people have a better memory than others? Do people's brains work differently?
Hi, that's a very interesting question. So how individual are different, but it's as how our brain works, all neuron connection. In one person, individual neuron behave differently. And then that is more big difference between human to human. And also it's how to make learning is very different. Some people can really naturally coming in without effort can make memory, but some people have to spend the effort to make memory. So it's something connection, how to make it all individually. So it's a learning way is different. And when you try to remember something, how you concentrate all things. So example, when you early morning or late afternoon, if you compare yourself, it's sometimes you cannot really remember in the morning, but after a couple of hours coming back. So it's very different, but you could train to becoming more, it's easy to make memory by training. So that is a lot of this memory training and these things. And now very popular is using called place memory. So you make space or place in your brain, and you put your things you want to remember, you put in this individual place that you just have to train. That make you more easy to remember. So that is, so basically really network is so complicated. So even you cannot control, but that is really advantage of human being, because the different memory, different event, all things, you have many different area of brain habit. Someday you just have coming out many piece and piece, and you can be very creative. So that is good part. Bad part is that's not always coming. So that feel like I don't remember many things, but that's actually not the bad things. You have to be patient with your brain.
That's great, thank you. And we have another one for you on Facebook that's just come in, which says, what are the differences between animal memory and human memory, and how can you tell?
I see. Well, I think big difference is human and animal. Is well, even human, it's all that's ancient type of human before culture developed, and the current human it's different. So basic of survival is more important than animal, and human used to survival was more important. So it's much more closer to the animal. But now, after this modern time, we have a more developed cortex, and not only survival, but its memory mechanism itself based on survival. is a basic. So that's, you have to train your brain to remember something. This is related to survival. But passing a test is if you can think about survival, but kind of. But that's basically different. Survival and how to keep your race for as the next generation. That is most different. But human beings went to further step. That's different. But the basic mechanism to use this synapse and neuron connection are the same. So basically principle how to make. But the part is a more important, necessarily it's a different between human and animals.
Great, thank you. And we've got one for Inga, which has come in on Facebook as well, which says, how do you treat the chordoma?
Hi, thanks for an interesting question. So my background is surgical, which is why I'm interested in chordoma, and the primary treatment, the main treatment is surgery. There are no drugs which can be used to treat chordoma, and you can also, after surgery, most people have something called proton beam therapy, which is basically a very accurate form of radiotherapy. Because chordomas grow very slowly, they're not very sensitive to things like chemotherapy, and they're not very sensitive to things like normal radiotherapy, so you have to use special treatments. But it is mainly surgery, which is why we're researching hard to try and see if we can develop anything else to treat it.
Great, thank you. And I think we'll give you one from that we had in advance as well, which is, why does chordoma grow only in certain places like the spine?
That's a good question. So it's one of the reasons that we think chordoma is related to the notochord, or one of the pieces of evidence that links the two things. So chordoma almost exclusively grows in the spine or in the base of the skull, and this is where the notochord was in the embryo, if you remember in the video, the structure that it looks like. So we think that's one of the links. There are some rare cases where they grow in the lungs and no one quite knows why yet, and that's another area of our research.
Thanks. And one that's related, why does it take until you're 40 or 50 for the cells that haven't specialized to become cancer?
That's $1 million question. Yeah, so that's one of the things we're trying to find out. And our theory essentially goes that islands of notochordal cells probably remain after birth, when they should generally disappear and end up in your discs between your vertebrae. And we think that over time, like other cancers, that these cells then get mutations, acquire mutations, and then it's these mutations that drive it to become a cancer. We think these islands of notochordal cells, probably a fifth of people have them, just walking around, and they're fine for the most part, nothing happens with them. But that's our theory as to why it presents or occurs quite late in life, although some children do get it too.
Oh, great, thank you.
Thanks.
And we've got a question for Laia, which has come in, which is, apart from a car, where else would you find nanoparticles?
There are nanoparticles everywhere. There are nanoparticles that help cloud formation in the sky, because there are dust nanoparticles that allow water particles, water molecules to come together, and this is how you create a cloud. There are also nanoparticles used in the cathedrals, in the Gothic cathedrals almost 1,000 years ago in Europe, where we have discovered that there are gold and silver nanoparticles used in the big stained glass windows, that these create the different colors and the beautiful colors from the cathedrals. There are also nanoparticles in the space, in what is called the interstellar medium. And that's a very interesting topic of like astrochemistry to study.
Great, thank you. That's kind of tied into the second question, which was, do nanoparticles exist in nature, or what's their purpose? But I think you've kind of answered that.
Yeah, that's a very philosophical question. Yeah, nanoparticles exist just because the elements break into smaller pieces, and that's how you create them naturally.
Great, thank you. And I think we're going to go for a question for Eleanor, which is, what do you think is the most interesting thing that's going to be able to be done with quantum computing in the future?
Yeah, that's a really relevant question. So I think in the near-term future, most interesting well, there'll be a lot of materials discovery and drug discovery, and in the long term, maybe optimization problems. So with that, I mean, kind of every material that we have. So the wood that the table that I'm resting on now is made of atoms which have electrons and protons and neutrons, and the way that they interact with each other and also with all of the other atoms in the material, that's a quantum problem. So if we can understand that, and that's the same for drugs, so molecules are just basically assemblies of atoms, and understanding how the molecule acts and what's the trajectories of all of the electrons, for example, and where they sit and what the ground state is. These are problems which, if we can understand how all of the atoms work together and all of the electrons work together, then we can actually design better materials, which are maybe stronger or lighter or more efficient. Maybe we can get, I mean, a big problem, for example, at the moment that I'm also working on is high temperature superconductivity. So if we can make a material which conducts electricity at very low loss, we can just conduct electricity around the world, with nearly no loss. So once the infrastructure is in place, it's practically free to transport the electricity. And for long term, maybe optimization problems or linear, increasing, speeding up linear algebra would be useful for machine learning. And then we can understand maybe consciousness better, probably links in a lot with some of the work which people are doing here. So yeah, that's general.
It covers a lot of ground.
Yeah.
We've got another question which sort of touched on earlier for you, which is how big is a quantum computer, and can they be made smaller in the future?
Yes. So I realized this. Yeah, this did link in with earlier. Basically, no quantum computer implementation is probably bigger than the size of a room, and they all hold within more or less the size of a room. But it depends on the type of implementation. So of course, the quantum part of the quantum computer is absolutely minuscule. Generally, it's really kind of small. Is actually where the quantum stuff is going on. And that's one of the things about quantum is it happens at very, very low level, and you have a Bose-Einstein condensate, which is called, and that's one of the largest quantum entities. It's nearly 1 micron in size, but that's the biggest quantum thing which exists. Well, with the biggest entirely quantum entity which exists. So how big is a quantum computer? Maybe hopefully that kind of replies, because you also have to take into account maybe the vacuum chambers and all of the electronics that surround it, and often they need to be cooled down to very low temperatures. So that also takes a bit of space. Yeah.
Thank you. And I think we'll go back to Helen, please. Someone said, is trophy hunting a controversial issue among people who work in conservation? And do you have personal short thoughts that you'd like to share on it?
That's a tricky and good question. In terms of controversial in conservation, I guess it kind of depends on what your definition of conservation is, because it really does cover quite a wide spectrum. So there are organizations that are much more on the animal welfare side of things, where they're very against trophy hunting. But then you have organizations which work with people a lot, which are maybe a bit more, I don't want to say practical, but sort of maybe holistic and looking at the bigger picture. Then they generally sort of consider it to be one of the tools in the conservation toolbox that shouldn't not be used because it can do some good. In terms of my personal thoughts, they're quite mixed, probably more so after studying it for such a long time. But I think where it does work, or like there are some places where it does work, so there shouldn't be a blanket ban, for example, which is being called for. And it really needs a more nuanced approach. But most importantly, I think that it shouldn't be up to someone like me to decide. It should be people who are going to be involved by affected by those decisions that should have the most say in what happens.
Great, thanks, Helen. There was one which is now quite far down and I've lost, but it was something along the lines of what are you planning next for your research, where to take it after this?
Oh, that's quite interesting. Well, so my research has had to change quite a lot because of COVID. I was meant to go back to Botswana for another 4 months last year, to do some work comparing villages where there had and hadn't been trophy hunting, and sort of looking at a before and after picture, so that you can really quite accurately, or like more accurately, tell what changes were down to trophy hunting. So if I did carry on with this sort of work, I would probably do something like that to get a better, like really quite solid understanding of how that change happened.
Great, thank you. And we've got one for Inga. How do cells in an embryo know what parts of the body they're going to be?
This is a great question again. I wish I had firm answers, but I guess we wouldn't be doing this research. That's a fantastic question, because if you consider some of what other panelists have talked about, of course, all of our cells have exactly the same 3 billion base pairs of DNA. And yet from this same DNA, from this same recipe book, we make all of the plethora of cells that make up our human bodies. So that is a good question, and there's a lot of research not only in the mouse but also in the human to try and map out what happens to cells. So quite an interesting question is, at what point do you know what you're going to become? At what point do you commit to a certain fate? So it's currently unanswered, but there are lots of interesting projects, such as one I'm sort of involved in at the moment, where people are trying to create an atlas by doing sequencing on individual cells and putting together tens of thousands of cells to try and answer that question.
Great, thank you. And there's another one for you here, which says, is there any way of preventing chordoma or identifying people who are vulnerable to getting it?
We do know that there is a genetic, it's not a mutation, it's something that you inherit, but it's basically very common. So it's present in half of the population, but of course, it's a very rare tumour and half of the population do not get a chordoma, thankfully. So we know that it's part of the puzzle, but not the whole story. So it's probably a series of events, and it's probably quite a complex process. So we don't know the full answer at the moment. It's probably a few things, like many cancers.
Great, thank you. And we've got one for Mina, which says, why do you think patient-centric medicine is important?
Very good question. I think it's my first answer would be around, the patient again. It is about really maximizing the therapeutic outcome for them, because we would like to achieve better and better therapies for everyone who are suffering from different disease conditions. But when we start, synthesizing a molecule and then designing into a medicine formulation and then running some trials to understand the performance of these medicine systems, this is about the medicine itself. But on the other side, we need to think about how the patient is basically responding. And there is a two-way, basically, direction in our research. Like, we are trying to make sure that we are designing and using the best scientific tools and models and understanding and know-how possible technologies to make the best therapeutic approach. But on the other side, we are trying to keep our focus on how the patient is actually treating, this medicine in their body and how they are responding, as I said. So I think this is very critical to ensure ourselves that we can expect what we designed to, achieve in this journey. So I think if we try to understand that if the patient is responding to the treatment in the way we predicted, also if the patient is being able to take the medicine in the way we prescribe, these are two very important factors, because I haven't touched base in the Q&A session so far, but the administration of the medicine is very important. And I think it ranges from different elements, from medicine related devices to digital related interventions to behavioral medicine to adhere to their treatments for a longer term. So I think this is very important to hear and understand, what are the individual needs of the patients for taking their medicine for a long time. So are they suffering, at the point of administration? If you go back, a tablet or a capsule to be, taken with a glass of water, can they, swallow it easily? Because we would like to avoid any medicine-related errors, any risks because of incorrect use. So this is very important to understand on a daily basis what they are suffering from and how we can really maximize the setting in the best way for them to ensure that when they leave the GP and pharmacy, they understand how to take their medicine correctly and then accept this medicine because this is for their treatment, and adhere to this treatment for the required time for this medicine to be taken.
Great, thank you. And I think we'll move on to one for Vanessa, which says, how do you study the diseases that you discuss? Do you work with people with these illnesses?
Yes, so our approach is really we want to understand the genetic basis of life-threatening infections. And we do this in humans. And what we do is we, in a sense, take advantage of these experiments of nature, horrible experiments of nature, the unfortunate occurrence of these exceedingly rare children presenting with life-threatening disease. And we focus on them and try to understand what it is that went wrong. So we basically recruit patients with these rare diseases. For example, the example I've used is we work with herpes simplex virus 1, or HSV1, which causes a common cold sore. We are actually interested in looking at children who present with brain infection caused by HSV1, which is very rare, occurs in 1 to 2 cases per million per year. So this is exceedingly rare. And so by recruiting these patients, we study their DNA, we study their cells and how they respond to the virus in a test tube. We try to match up pathways and identify genetic changes that could possibly explain this. So this is the approach we take, is basically we recruit these patients and then we genetically sequence them and also study their cells' response to infection.
Great, thank you. And we've got one for Heidi, if we could go back to you, please. What kind of skills do you need to run international conservation projects? For example, is it difficult to collaborate with researchers who are so far away and speak different languages?
Absolutely, and that's something I often think about myself, and I try to reflect a bit and also discuss with my colleagues. So for my work, yes, on a daily basis, I do work in both English and Chinese. So there are language skills depending on in what countries your collaborators are based for the most part. So being bilingual has definitely been, I'd say, one of the biggest advantages, or rather requirements for my current role. But otherwise, I have to multitask a lot. So that's something I still try to train and practice in myself, since I'm managing everything from budget to logistics when we travel and go to the field, do field work, or even have meetings, to actually doing the research, working either with my team here in London or with our collaborators or students, and doing the data analysis, writing papers, and as well as other more fun, interesting things like public outreach, for example, that require a different set of skills. But I would say, yeah, multitasking is another big one. And I'd say also just having lots of interest is also something I see in a lot of my colleagues who do similar types of work or coordinating big projects across sometimes 2 or even more countries. It's just always having an open mind, talking to different people, hearing their opinions, because culturally things can be very different across societies. So I think that's also something that, I constantly try to remind myself as well, is to be open-minded and really respect and listen and try to incorporate different views. And I think that also brings a lot of value to our projects. So yeah, I think language skills is one, multitasking is another, and just keeping a very open mind and curious kind of mentality is another one.
Thanks. We've got another very quick one for you, which is, where can people find out more about animals from Hainan?
Sorry,
where can people find out more about animals from Hainan?
So I would love to be able to point you to one specific place, but in my knowledge, that doesn't really exist. And it's something we would love to promote a bit more. For example, like events like this, because first of all, China is very, very diverse. It's a continent's worth of different environments, different species, different regions. And that's why I talked about Hainan in my presentation, because it is a place that's quite unique, but maybe not that well known, definitely not as much as giant pandas. So I would say if you or interested in any of the species that I talked about or anything related, I think looking up the species themselves might give a bit more information. So for example, the Hainan gibbon now is increasingly better known, I think thanks to a big network of partners that we've worked with. The big-headed turtle, for example, is an EDGE species, so that's got more coverage now. So if you go on the EDGE website, there's a specific page dedicated to the big-headed turtle, and not just in Hainan, but also in Vietnam, for example. So yeah, I would say look up the species and you'll find lots of other individuals and organizations working on them besides us at ZSL. So.
Thanks, Heidi. We've got one for Zoe, which says, this was one we got in advance, saying, your video was beautiful. Did you draw it yourself? And do you think art and science work well together, even though they're traditionally seen as opposites?
Thank you for the very kind words about my video. I did draw them myself. I have a tablet-like computer that I can draw directly onto, and I've always been somebody that's at the interface between art and science. And I think one of my music teachers said that I seem to be like a woman of the Renaissance, which is quite funny. And I think there should be more of mixing the two. I certainly enjoy it, and I thought I had to choose when I was younger. I thought I had to choose between art and science, and I did choose science, but I've always been interested in things that look pretty as well. So yeah, we should do more of it.
Great, thank you. And we've got another one which says, how do you know DNA comes in different structures? Can you see them under a microscope?
Yes, that's a really good question. We use lots of different techniques to look at the different types of DNA structures, and you can see them under special types of microscope as well, using atomic force microscopy and things like that. We use a lot of light-based techniques to look at the different types of structures, because they absorb light in different ways, and that's the main method that we use to look at them. Another method that we're expanding into in collaboration with other colleagues in the School of Pharmacy is looking at crystal structures and actually visualizing that in a different way too. So lots of different ways.
Great, thank you. We're nearly out of time. We've just got, I'll ask one last question for Sheila. Are there things you can do which in particular are recognised to cause ovarian cancer? How many women have ovarian cancer without being aware of it? Does it always have severe implications?
Okay, and sorry, could you just repeat the first part of the question?
I said, are there things you can do which in particular are recognised to cause ovarian cancer?
Okay, and so in terms of that, I'd say the answer is generally no. There's nothing you can specifically do to cause ovarian cancer, unless, of course, you're being exposed to X-ray radiation, or you're working with carcinogenic substances, which I highly doubt many of us are working with in this day and age, and we're quite protected from that. So I wouldn't, and if I'm to go back to the nature-nurture debate, actually, I'd like to revise my answer to that and say that the majority of the diseases is an interplay of both. So what happens is your genetics tend to predispose you for a given condition. And if your lifestyle is poor, that will just tip you over the edge, and then that's when you'll get the disease. So some people may have certain genes which may increase their risk of getting ovarian cancer, but whether or not they get it may also depend on how their lifestyle goes, for example. And there are certain things that are known, such as, for example, having high sugar intake in terms of your diet, for example. Of course, I mentioned exposing yourself to X-rays or such kind of radiation unnecessarily, that can also increase your chances of being exposed to carcinogenic substances. And the second part of the question?
It was, how many women have ovarian cancer without being aware of it, and does it always have severe implications?
Yeah, so that's a very, very good question. So first and foremost, there are many types of ovarian cancer, just so you know. And the particular cancer that I look at is known as your high-grade serous ovarian cancer. So this is the most lethal subtype. And in terms of how many women have it without knowing they have ovarian cancer, I'd say a lot. I can't give you an exact statistic of how many who have it and don't know, but it is a lot of women. And the reason being of how many women who have it and who don't know about it is a lot of women. And the reason being is because ovarian cancer is found internally. So it's really hard for you to feel that lump and know that I need to go and see a doctor. I have a lump in my ovaries. I don't think any of us can feel our ovaries. So what happens is we tend to catch this later on once we've had other symptoms such as fatigue, for example, and some of these symptoms can be passed off as having another disease. So it's just later on we tend to pick up on this, and that's where my cancer comes in, high-grade serous ovarian cancer. So at this point, you start experiencing these symptoms after the cancer has metastasized, has spread to other parts of the bodies, such as the omentum, and it's actually very lethal. So what I can tell you is approximately 83% of women with ovarian cancer do die from the high-grade serous ovarian subtype. So that's why in our research, we are trying to find biomarkers such that we're able to try and detect it earlier before it has metastasized, where you can't really treat it with chemo, you can't really salvage it with chemotherapy or maybe surgery. So that's what we're now moving towards, more preventative measures.
Thanks, Sheila. So we're almost out of time, so I'm going to ask one last question, which is for everyone. Which someone has said, what advice would you give to a young person interested in a career in science? Maybe we can start with Sheila, as we just had you there.
Yes, okay. I'd say for a young person who's interested in a career in science, I'd say what you're doing right now is absolutely great. You're on the right track. You're attending such events. You're getting to know more about science. I'd say at a young age, it really is more about passing your exams and ensuring that you do get onto you do get to do your STEM subjects at university, as they tend to have a higher cap compared to your non-scientific subjects. So I'd focus a lot more on really just achieving the best grades you can, and maybe also trying to diversify if possible. I wouldn't advise you to limit yourself, as I think it was Zoe who mentioned about being at the interface between art and science. Even within the sciences, like if you pick chemistry, you can easily pivot towards biology. And say if you pick maths, you can easily pivot towards physics. So maybe just try and diversify as much as you can.
Thanks, Sheila. So maybe we'll go to Zoe next. Have you got anything you'd like to add?
Sorry, turning my camera on and off. Yeah, so I would say that if somebody tells you you can't do something, then just go for it anyway. There's so many barriers in life, and don't put anything in front of yourself. But if somebody tells you you can't do something, that definitely doesn't mean you can't do it. And I know it's sometimes difficult to pick yourself up from that, but you can be very much determined and still get there. So that would be the advice that I would give. Thank you.
Zoe. And I think, how about Ella?
Yeah, I would say the most important thing is to try and get involved with as many things as you can. So see if you, depending on what level of education you're in, if you have any kind of teachers or university professors, just go and ask them, is there any kind of project that you can do? And really importantly, depending on, if you haven't started a PhD yet, there's lots of different areas you can go in. And the important thing which people who are offering PhDs are looking for quite often are, are you good at being able to solve scientific problems? So if you have good grades, you can actually go to many, you don't have to continue in the direction which you started in your studies. And so it's really important to make sure you're choosing the right area early on. So yeah, as people have previously said, continue to attend these events so that you can actually find out what area you're most interested in. And when you think you maybe have a good guess at what area you're most interested in, if you can get involved in any kind of projects outside of university, considering that you're doing fine in school and university, then yeah, really go for it, because that will make you stand out at the later stages. Yeah, and keeping team, I think, is the most important.
Thanks, Ella. How about Leia?
About what topic would you like to learn, everything there is to learn about it, or what topic could you talk about for hours, or what problem or scientific question would you like to solve, and that will give you the motivation.
Thank you. How about Inga?
If you can, I think it's always possible, is try and find yourself some kind of a mentor. So either through things like this, I feel like people are always so willing. It's amazing how willing people are to give you a little bit of their time to guide you in a direction that you're interested in. Particularly if you say, you know, essentially, I want to do something like you do. I think everyone has always got time for that.
Fantastic, thank you. How about Keiko?
Yes, I would like say also try to, as early as possible when you start interested in science, to make good contact or any kind of experience you can get it. Just start small things start, and then from there to expanding to the opportunity to experience how you like it. I think you like it on actually doing very different things. So it's you just try to taste it and you feel it, and then from there to go.
Thank you. And then we've got two more I'd like to ask of, maybe Heidi.
I think for me, in addition to all the great advice everyone's said so far, is to look for the science just around you in everyday life. Science is, you know, not just in the lab, not just in the classroom. It's really everywhere. So being, just being aware, being curious, ask questions, and if something just really fascinates you, then maybe try to read up about it, or ask someone. And also just trying to explore the opportunities around you. For example, museums are a great place to start. They often also have events for like talks and meeting scientists and meeting people in the field. So try to take advantage of these. And also just being well-rounded, I think, is another important thing for myself as well. Is really, even though we work in science on a specific discipline or a specific sector in science, a lot of these issues we're dealing with definitely have major societal implications, ethical implications, and just, not just studying the science, but also the place of the science and its relationship with society, with people, with all the other things that science is part of in our world.
Thanks, Heidi. And lastly, Helen.
Yeah, I think I'd probably say it's to definitely keep an open mind. Science is so broad, as these talks today have demonstrated. It's sort of, if you try one thing and think, oh, that's not for me, definitely don't give up on science as a whole, because there are so many different avenues that you can follow through and try out.
Great, thank you everyone. Thanks for your great advice. And thanks everybody who joined us on Facebook. Thank you for all your amazing questions. If you have any other questions that didn't get answered, you can always tweet us and we can see if we can help you with that. And thanks again to all of our panelists for your amazing videos. Thanks and bye.